Good afternoon, everybody. My name is Tony. I'm from a company called MEDIPOST. Most of you probably have not heard about us, and then we're a Korean company, a small Korean company established in year 2000. If you can move to the next slide. We were established in 2000. Started off as a cord blood banking company. We still operate the largest private cord blood bank in Korea. With the parent company is a public company traded on Kosdaq with $370 million market cap right now. The therapeutic development and product marketing is focused on the Asian region. Its own subsidiary, 100% owned subsidiary and based in North America. That's where I lead our North American activities, as well as from next year we'd like to begin our European activities. So since the beginning of the company was 2001 to for about 10 years, the company has been doing R&D work using mesenchymal specific populations of mesenchymal stem cells or stromal cells derived from umbilical cord blood as a concept of a allogenic, which is quite opposite to the autologous. As we've heard from the previous speakers, we're in the business of developing allogenic donor cells sourced from umbilical cord blood, which is donated, otherwise would be discarded as medical waste. So manufacturing standard procedure is we get the donated cord blood unit, specific cell population has been isolated and then separated and culture expanded at a certain stage where the what we call drug substance or the aliquots of frozen cells can be stored and frozen for up to a number of years as approved by regulators. And the concept is then those pre-manufactured off-the-shelf products or stocks can be thawed and then suspended and into a final product or vialed and then released into clinics. And the one lead product that we have developed and have finished phase 1, 2, 3 clinical trials in Korea and has been on the market for the last 2 years as an allogenic off-the-shelf product. It's called CARTISTEM. It's for degenerative osteoarthritis or cartilage defect. I'll show you some data on that. It's the same product has been approved for clinical trials, phase 1, 2 trial by the US FDA, and that's currently undergoing a trial in the US. So mechanism of action, as we heard from excellent previous speakers, is paracrine. So these cells, when they're introduced into the defect area of the host, where the chondrocytes of the patient's own cells are still there, but these cells, when they're introduced to the environment, they detect the signals that are present in that disease environment and then secrete other proteins or paracrine factors into the area, which causes the stimulation of endogenous regeneration as well as very efficient inflammatory reaction modulation and management. Then after a certain period of time, these cells basically die naturally and then they get digested. So through this paracrine mechanism of action, we've done extensive preclinical for safety and efficacy, and the product has been developed. As you see on the top picture there, for the contrast reason, we've got the red color in that cell suspension. At the bottom, we've got cells, and then the product has second component, which is freeze-dried, high molecular weight, hyaluronic acid that gets mixed together just before the administration into viscous gel, and through an open surgery or arthroscopic surgery, it's directly applied to cartilage defects, such as shown on the top bottom left corner there, which is one of our patients that was in phase 1/2 trial. You can see the long-term follow-up data that leads to the self-regeneration or endogenous regeneration of the patient's own cartilage. So cartilage defect can be subdivided into 4 categories that are determined by the ICRS, which is Society for Cartilage Repair. And basically, the grade 1 is superficial loss, and grade 2 is less than 50% of the thickness of the cartilage loss, 3 is more than 50% loss, and 4 is complete loss of cartilage, exposing the bone. And through the severe forms of cartilage defect, as in the cases of 3 or 4, their current standard care is what's known as microfracture, which is basically drilling through the holes of the bony area and then causing the bone marrow to bleed down from underneath the patient's own bone marrow. That's all you do, and then the stem cells from the bone marrow does lead to some regeneration, although it is limited by the patient's age. The other alternative extreme end of current interventional care is artificial knee implants, or the total or partial knee replacements, where the artificial kneecaps have been put on to these cartilage lost area of the bone, and that is a very invasive, but also is limited by the age. As shown in this graph, which is a 103-patient randomized controlled trial phase 3 that we've conducted in Korea now some 5 years ago, that has a specific population of subjects that we've enrolled, represented by the yellow bars, in those age groups, mainly focusing between roughly 50 to 70 years of age. The reason being the microfracture, which is currently available, is limited by patient's age, that it doesn't lead to a meaningful regeneration of cartilage if the patient who gets treated with microfracture goes beyond the age of 50, 55, very well known in the community. Whereas the artificial knee joint joint replacement surgeries are not recommended patients until they're old enough because of their limited lifespan on the actual implants. So when we enroll this pivotal study, or subjects for the pivotal study, we really focus on that age group by using allogenic cells that causes the self-regeneration of cartilage, where we can bridge that gap as currently unmet medical need. And obviously, if we can do that for those patients, then that same cell or the product should work across the wide range of age groups. The other limiting factor in the field of orthopedics is that the microfracture, even if it works in the young patients, only work for a very small size, relatively small size of defects, roughly sizes of 3 to 4 square centimeters at most. So in the enrolled patients, because they suffer a general OA as well as in the older age group, the patients, as you can see in the distribution of the size of the defects that we've treated, goes as large as 8 to 9 square centimeters. So this was another challenge that in the field, it doesn't matter which current intervention care they choose, unless it's total replacements, the methods that are available are really limited by the size of the defects. So what we looked at then is, after treating these patients either with microfracture or our cells, randomized, and looked at 48-week time point, and then did a blinded assessment by an independent orthopedic surgeon of the regrowth of this cartilage. So all the patients enrolled and treated were at grade 4, which is the bare bone stage, and then at 1 year time point, as you can see on the left-hand side pie graph, we examined for the growth of the cartilage. So the blue pie, which shows 2%, they didn't show any regeneration, so they were grade 4 at the time, which is the same status as when they got the treatment, as opposed to 28% on the microfracture on the right-hand side graph there. And you can see the green and then the red and then blue, which gets to grade 3, 2, or 1, which is significant improvement or regeneration of cartilage, and that is some 98% on the CARTISTEM case, whereas that is less than 70 or so % in microfracture. The other really important aspect, if you follow this area, is the type or the quality of cartilage that leads to regenerate after any treatment. And in this case, microfracture, you can see the young patient who was treated with microfracture with 2.25 square centimeter defect at time of treatment. This is obviously 1 year after the treatment. You can see the white cartilage through the arthroscopic image here, but in the biopsy samples through histological staining, what that shows is lack of natural ingredients, if you like, the components of the hyaline-like cartilage that should really be in the natural status. So what that represents is, as well as very well known in the literature, microfracture, even for young patients, that leads to cartilage regeneration, but it regenerates the wrong type of cartilage known as fibrous. Whereas the CARTISTEM treated patients, despite the age, in this case 65-year-old female, it leads to cartilage regeneration, and through histological analyses, through biopsy, we were able to biopsy every patients on this trial at 1 year time point. It shows that a very natural hyaline-like cartilage in their quality. So this was another very encouraging results that we were able to follow. So we've just finished 3-year follow-up data of the same patients after single treatment last year, and this year we're in the middle of doing the 4-year follow-up. And this was data from last year. We've just finished analyzing earlier this year, which includes a long-term safety examination using MRI images, obviously through any potential safety concerns long-term by using allogeneic cell transplant as a product. There was no safety concerns whatsoever, but more importantly, we were able to look at some pain and functional scores of these patients after single treatment at 3-year time points, which is last year, as I said. You see this graph here, it's a representation of scores by measured through WARMAC scale, which is a measure of functional scoring, and the lower the score, the better. So the far left dot is at the time of treatment, the red line indicates the microfracture treated group, and the blue line is a CARTISTEM treated group, with the both of which improve by decreasing in the score at 1 year time, which is the middle point. And then if you follow to the far right point, that's a 3-year time point where the red line, which is microfracture group, treated group of patients, the score gets back to the baseline, and the blue line stays on as improved score at 3 year. And the same, a similar measure of functionality by IKDC scoring shows a very similar pattern where in this case, the higher the score, the better. So the blue line goes up, red line goes up together at 1 year time point, but the more importantly, at 3 year time point, the microfracture treated group, the far right-hand side, deteriorates back to the baseline or state where the time when they're getting the treatment, whereas the blue line continued to show an improved state of functionality. And this was a very important confirmation for us, not only through morphology that we were able to confirm at 1 year time point, but the functionality can sustain the improved state. So one quick on-market study, as I mentioned to you, this is now available in Korea. So this particular patient in his late 50s received 4.5 years previous to this image of microfracture standard care, and with a pain score of about 65. The 100 millimeter VAS is a visual analog scale of a pain measurement, very subjective scoring, a 0 being no pain, 100 being unbearable pain. So they pinpoint a number. So 65 was what the level of pain that he was feeling. After second microfracture, it was still showing lots of red areas or bony areas and a lack of cartilage. And then the pain score was around about 650. And then this patient received CARTISTEM treatment, and following 6 months, you can see the regeneration of cartilage on the arthroscopic images, all the white portions there. But more importantly, the pain score was reduced right down to 5. Now, so these are types of on-market data that we're getting now from the market, and this particular product has been developed with Samsung Medical Center Orthopedic Department. And this has been labeled, as I said, for the repetitive traumatic cartilage degeneration or defect, including degenerative osteoarthritis, more importantly, without age limit, and because of the inclusion of those aged population subjects in our pivotal study. So the Korean market status as of this month, we've treated over 1,500 patients on market, and it's available across the country through clinics and hospitals. The Korean FDA has mandated us to do 600 patient post-market surveillance, which is being carried out right now. We're at the very final stages putting a market approval application for the Korean dossier, basically including all the technical, CMC, and preclinical, as well as our clinical trials data for 2 regulatory jurisdictions, one Australia and another one Canada. We have a very strong focus in the past few years in North America. And to just give you a glimpse of what the market size we're talking about here is, this is data from Arthritis Foundation, Alliance Foundation from Canada, shows some 35 million population of Canada, some 4.5 million Canadians have OA. And then this number is going to go up to 10.4 million by 2040. Now, expected population in Canada by 2040 is about 40 million. So this is 1/4 of the entire population. And you can see this, the increase in by decade, the lines, different colors, is really showing that age group of patients who is going to experience the most increase in terms of the demographics is that between roughly 55 to 70, as I showed you in our pivotal study design. With that is, there's no current intervention care. Apparently, there hasn't been anything in the last 20 years that can really potentially help those patients. And that's why we think by demonstrating through our clinical pivotal study that the CARTISTEM can really link that so-called unmet medical need, and this population in the market will ever going to increase with the aging society and so forth. So the last one quick slide is that we've got 2 other products in the pipeline apart from CARTISTEM. The other second in line is what's called PNEUMOSTEM. This is for Broncho-Pulmonary Dysplasia, which is for prematurely born babies, and they are born at 25 weeks gestation, cannot breathe in air. We deliver cells straight through the airway. This has been completed phase 1 trial in Korea. In fact, this was published in the Journal of Pediatrics a couple of months ago. We are almost finishing phase 2 in Korea with that product, and also have cleared a US FDA IND for phase 1/2 trial, which will begin in the US next year for the lung product. Very lastly, very quickly, and the Alzheimer's disease program is in phase 2A trial in Korea after successfully finishing phase 1 on mid to late stage Alzheimer's disease patients last year. So the business model is that the Korean company has opened up a subsidiary in North America where we established and we have exclusive commercial rights to the older jurisdictions of American continents and Europe, as well as Australia and New Zealand. And we will be, as a private company, will be opening and offering a private placement for this US or Canada entity, or the North American entity, I should say, for those regional rights and expansion into European regulatory jurisdictions, with which we plan to initiate the discussions of which phase of trials that we can enter into through EMA beginning next year, and that will coincide with our private placement of next year. Thank you very much for your time, and here's my details for your contact.